My name is Murali Gopalakrishnan, and I head up the global search and evaluation function within the corporate strategy office here at AbbVie. And on behalf of my co-chair, Tim Luker from Eli Lilly, it's my pleasure to invite and welcome everyone to this panel entitled Next Generation Neuroscience, Tackling Neuroinflammation and Degeneration. And as all of us will agree, this is a very timely topic. And as Tim explained and highlighted, we are seeing a renaissance in neuroscience today, and we will all discuss that today, and we will, as a panel, go through the emerging trends, the opportunities, the challenges, particularly in the context of neurodegenerative diseases. And before we jump in, let's go ahead with our customary introductions. We're going to start with my co-chair, Tim Luker. Tim, do you want to start from your side?
Murali, thanks for the great intro, and obviously thanks to Sachs and the organizers for putting this exciting panel together. My name's Tim Luker. I'm a VP in Corporate Business Development at Eli Lilly. It's a major pharma company. We have extensive interests in neurodegeneration and chronic pain. We have exciting products coming, and it does feel to me like there's a big renaissance in neuroscience at the moment. So really looking forward to debating that with the rest of the panel today. Maybe I could pass over to Deborah to introduce herself.
Yeah, just as we introduce for the rest of the panel, just a quick reminder, just mention your company, obviously, and the lead program, the lead technology that you're pursuing, and maybe highlight the general strategy that you're thinking about therapeutics or drug development in the space of neurodegeneration. That will be helpful. Deborah, go ahead.
Thanks, Murali, and thanks, Tim. So I'm Deborah Rathjen, and I'm Executive Chair of Bioasis. Bioasis has a platform technology for delivering biologics across the blood-brain barrier. This proprietary technology utilizes a 12 amino acid peptide and a receptor called LRP1. There's a robust patent portfolio that supports the proprietary nature of the platform technology. And for our internal pipeline, we focus on the intersections between lysosomal function, activation of the inflammasome, and neurodegeneration. We have programs that are targeting replacement of progranulin and replacement of glucocerebrosidase, for example. So covering lysosomal storage disorders and then into neurodegenerative disorders that are associated with genetic mutations.
Thank you. Thanks. Bharat.
Hello, everyone. Good morning from Boston. My name is Bharat Ramachandran, and I work in the business development and licensing team at EMD Serono, which is the US affiliate of Merck KGaA Darmstadt, Germany. Within the business development team, I've been in this role for the last 6 years. Focusing more on the licensing and the transaction side of things. At Merck, we focus in neurology and immunology. These are really broad areas. So our focus on the neurology space is more on the neuroinflammation aspect, and in immunology, it's more on the autoimmune diseases. In terms of our pipeline, we're really excited for evobrutinib, which is a BTK inhibitor that is in phase 3 development for relapsing forms of multiple sclerosis. And we also have 2 other marketed products in multiple sclerosis. Excited to be here. Thank you.
Great. Thanks, Bharat. Jesús, do you want to start? You're muted. You may have to unmute.
Yes, hello, I'm Jesús Martin-Garcia. I'm the founder and CEO of GeNeuro, which is a Geneva, Switzerland-based company. We are a phase 2 clinical stage company that develops antibodies to neutralize patient-produced pathogenic proteins that fuel a great number of neurodegenerative and autoimmune diseases. These pathogenic proteins come from the 8% of the human DNA that has a viral origin, which are human endogenous retroviruses of herbs, which are normally silenced but may express such proteins when viruses from the environment interact with human cells. Our lead candidate, temelimab, is finishing phase 2 at the Karolinska Institute in Sweden with the results in March next year. We're very excited. And this technology is also, we're seeing applicable to inflammatory psychosis, long COVID psychiatric syndromes, where we see the same mechanisms at play. And we're also active with the NIH against the ALS. We have a partnership with the NIH against ALS, with results that will be presented at the ANA in a few days, actually.
Thanks.
Very excited to be in the panel. Thank you for the opportunity.
Great. Thanks. And last but not the least, John.
Thanks, Murali, and thank you actually to you and Tim for inviting me to speak at this panel. I'm very much looking forward to it. So I'm founder, CEO of BIP Pharma. We're a Boston-based private company in the CNS space. We are firmly going after tackling neuroinflammation, but we have a very specific take on it. We're not, in the traditional sense, an anti-inflammatory, in that we're not trying to reduce inflammation, rather going after the effects of inflammation, neuroinflammation on synaptic and neuronal function. Comes out of an observation a few years back in aged rats, where our drug that we're developing, a very specific oral kinase inhibitor, actually reversed fully the deficits in the Morris water maze, but it wasn't correlated to inflammation, but rather to inhibiting IL-1 beta signaling. We've run with this over the last few years and demonstrated that the effects of our drug are in terms of neurotrophic signaling and reversing deficits in cholinergic function in preclinical models. And then more recently, the very specific disease where cholinergic deficit dominates is in dementia with Lewy bodies, where we've reported positive phase 2 clinical data. We've been to the FDA. We have a defined phase 3 study. And ahead of that, we'll be working on starting up a phase 2b study to confirm the findings in the phase 2a study. We'll actually be presenting more data on this at CTAD in the coming months.
Great, John.
In early November.
Great, John. Thanks very much. Thanks for the overview. I guess we'll jump right into the topics today that we would like to cover. They're outlined on the slide. I'm not going to go through specifics of this, but obviously we'll start with Alzheimer's being the indication that sort of like to sort of begin with, and then certainly we'll touch on different aspects of that, the different approaches that are being taken. We'll touch on neuroinflammation. So John will come back to you to get more thinking around neuroinflammation. We can also talk a little bit about beyond Alzheimer's, because usually we tend to ignore all the other neurodegenerative diseases, but I think we should touch on some of them. Multiple sclerosis obviously being one of them, and ALS as was just mentioned. We'll also then switch to new modalities. Certainly, biologics, small molecules have been sort of the front runners, but as you've seen, a number of other approaches are being pursued, which are interesting, but of course challenging as well. And then we'll end up with what's going on in the partnering world, especially when we are not face-to-face in meetings, we are doing virtual meetings. What is the partnering landscape today? And of course, what is the investment climate? So those are the general topics we'll try to cover during the session today. So let me jump right in. Maybe the first one, as we think about key developments in Alzheimer's disease and neurodegeneration, the two obvious ones that jumps, at least to my mind, has been, of course, the conditional approval of aducanumab. And the second one that sort of has been sort of under that new spectrum has been sort of the evolution of the anti-tau antibodies, particularly the unsuccessful trials of the N-terminal antibodies. So maybe we'll start with the A-beta topic, and certainly keeping all the controversy aside. And the question for the panel today is, how has the conditional approval of aducanumab sort of changed the landscape in AD particularly, and perhaps broadly in neurodegeneration? And there are sort of three parts to the question. There are, of course, additional antibodies that are advancing forward. About 3 or 4 A-beta antibodies are now marching ahead through various stages of development, including regulatory reviews. The perspective I'd like to hear is, do we have the optimal profile yet? Or if not, what would a successful optimal A-beta antibody approach offer? And secondly, as these therapies become available, how is the landscape going to change? And it'll also be helpful to understand, what is the landscape going to set for other therapeutics that are moving ahead in this space, and also in terms of, the diagnostics landscape that may be evolving. So maybe I'll stop here and perhaps, I don't know who wants to take the lead on it, but, unless my co-chair has an immediate comment from a pharma perspective, maybe I'll let Tim comment, but then maybe move on to Bharat for his comments, and then we'll open it up for the rest of the panel. Tim, do you have any immediate comments on this?
Yeah, I mean, obviously I won't be talking in detail about, Lilly's development products. That's a little bit of a no-no, but obviously it's an exciting time. We've had a very long commitment to this area with earlier programs in the clinic. It has been, a tough journey to understand this biology, and I can remember the early days where patients were going into trials without any imaging data, and it wasn't even clear that they were a real cohort of patients. I imagine back in those days we didn't have these imaging agents, and then there's obviously the link between behavioral endpoints and real pathology, and the time it takes to understand disease stabilization, and whether it it really is stable, or can it be even reversed, would be amazing. That would be the future, of course. And then I guess when I was learning early days, I didn't really appreciate how complicated aggregated protein biology really is. There's a quite, in some cases, quite a mixture of species. There are seeding mechanisms, accumulation mechanisms. There's different parts of the protein that you can target with drugs, and the subtleties seem to make quite a difference. So I know at Lilly we've been very committed to making a difference for patients in this area. It's been a long journey. Aside from the controversy around the clinical data, as you mentioned, I think it gives me some optimism that there might be something for patients not too far away, given the momentum. And then I guess finally I would say there's always space for sort of combination treatments, but right now I would give a fairly optimistic picture.
Great. No, that's very helpful. And just maybe, Bharat, if you have any other comments from a pharma perspective, and then we'll certainly go to the other panel members. Yeah, Bharat.
Sure. So I'll repeat the word Tim just mentioned, which is optimism. I think in this particular indication, there has been so much effort to try and bring medicine forward that clearly has an impact. And at least this is that one first step. And I think this will really drive optimism, and it will provide further opportunities for patients as we move forward. So in that sense, also big kudos to all the patient advocacy groups that have been pushing for at least something in this particular disease. The other aspect that probably was reading the other day on an article around how long it takes to get drug from a medicine from start to the finish line. And oftentimes we think about the regulatory approval as that finish line. And in this particular case, this is a very stark example that it's regulatory approval is just one step. We need to go further beyond that to ensure that every patient who wants to have that treatment gets access to that particular treatment. And that is really where we need to push. And that is so relevant in context of this particular approval, because last week I was reading that the Mass General Brigham system will not be providing this therapy. And that just tells us of how much work we need to do. Finally, coming back to the exact question that you had asked around how this would likely impact the other similar drugs of A-beta that are out there. I think it creates a focus on the biomarkers. I think we will see a lot more emphasis on development of these biomarkers. And to call that out, both on the imaging side, but also on the fluid biomarkers coming. I think we will see further development in terms of fluid biomarkers, in terms of saliva, exosomes, some others, to complement your traditional imaging biomarkers in terms of your MRIs and your functional PETs. And I think that will really eventually move this from a space where we are diagnosing these patients better to really trying to identify asymptomatic patients. So I think this has really opened, I think this is the first step that will really bring a lot more innovation in this space.
They're very helpful comments. Anybody else would like to add on to that from a landscape perspective and how it's impacting some of the work that's going on in your shop? Jesús, you want to start?
Yeah, I think that what has happened in Alzheimer's could also open opportunities into other neurodegenerative diseases.
Mm-hmm.
Because the difficulty in our area of neurodegeneration is that we do have biomarkers, but we don't have the precise exchange rate between the biomarker values and the clinical values. And this is something where, in other areas like cardio, et cetera, they have much more precise background on the exchange rate of this biomarker means this clinical consequence. In this case, aggregation and cognition, we know they're going in the same direction, but they're not necessarily, the precise exchange rate is so variable from one person to the other that it's going to be very difficult to establish. So if you go beyond Alzheimer's, this could also be applied to many other diseases where there's tremendous medical need, but today very difficult long-term studies to establish clinical efficacy. I'm thinking, for example, of neurodegeneration and multiple sclerosis. We have today 16 drugs that hit inflammation, but not much effect on the neurodegeneration for the patients affected. But we know that neurodegeneration is basically driven by things such as brain atrophy, black holes. It's driven by the loss of myelin integrity, all things we can measure. So, could we have in the future strong data on those biomarkers also be a sign of neurodegeneration, and therefore having provisional approvals based on those kind of biomarkers? This is it's a door that's been open. We don't know yet whether it's half open and just for Alzheimer's, or whether it will open up to the entire field of neurodegeneration, which in my view would be needed.
That would be great. Yeah, absolutely. John or Deborah, any other comments you would like to chime in here?
So yes,
go ahead.
Go ahead.
No, go ahead, please.
Okay, thanks. I would just add, I guess, that, still the correlation between what's happening and what we can measure in CSF versus what's happening in the brain, there is still, obviously PK-PD relationships that are not well understood. And, again, there's the blood-brain barrier. So at Bioasis, we've recently shown that an anti-beta amyloid antibody linked to our XB3 peptide has a 17-fold higher increase in terms of presence in the brain parenchyma in the cortex. So what would that mean for efficacy in humans is yet to be determined, of course, but there are still I think so many mechanisms for trying to expand that window of efficacy that haven't yet been evaluated.
John, do you have a short point, and then we'll move to the topic?
My comment would be that I think in the background, and I think neatly dovetails into this, there's been huge advances with imaging of various modalities of understanding the subsets within AD. And the reality is that AD, there's a very small percentage that may have pure Alzheimer's disease that the animal models identify. And my hope is that with the A-beta approach, we can identify actually who those pure patients are. And I presume that there's actually very significant and obvious efficacy within some subset. And then we can start teasing apart other mechanisms and which populations and how we actually combine. And I think Bharat's point in terms of, plasma biomarkers of amyloid and tau pathology, they've really moved quickly in the last couple of years. And I think that's actually one of the very exciting developments in the AD space that complements nicely the aducanumab approval. And the two things together is what I think really provides this understanding of disease subsets and mechanisms with how co-pathology plays out with the approval I think is what should provide all of us hope in the AD space.
Yeah, and that's exactly also applicable as we think about the tau space, as we touched on very early on. I mean, so far, the track record has not been great. 2 or perhaps 3, depending on how you look at 2.5. Anti-tau antibodies, at least the N-terminal ones, have not been successful so far, which comes back to sort of the comment that Tim made earlier with respect to, are we targeting the right species, involved in different stages of disease progression, and do we have the right antibody? So lots of questions are still open there. And lastly, is an antibody approach maybe right, but are we able to get enough levels of those antibodies to stop the phenomenology that we are going after? I think that's the other question. So I think these are all intertwined comments, and perhaps, maybe we can start. I don't know whether Tim, you're going to make a point here, but if you would like to make a point, go ahead. But that's the other question to understand: what are the lessons learned, and how does it impact the path forward as we think about tau-based therapeutics?
I think for me, it just we remain excited about the prospects for tau. It's a long history of data backing that up. But I think, as usual in science, that the devil's in the details. And obviously, my understanding is, many of the failures recognized a certain component of tau, the N-terminus, and they appear relatively similar. Some of them had already failed in PSP, so perhaps we weren't surprised. But there's a number of next generation antibodies coming that recognize different regions. They appear to, at least preclinically, look perhaps a little bit better in some assays. But aggregating proteins, whether you stop the seeding, whether you try to remove the aggregate, the fundamental mechanistic drug discovery, there's a lot of fine detail in this area that maybe on the headlines it's easy to gloss over. Yes, there have been some failures, but I'm still optimistic personally. I think in many areas, first gen efforts with everyone doing relatively similar things.
Right.
It wouldn't be a surprise if further understanding and more advanced therapies actually win. So I personally, I think that the jury's out, but we remain excited about this area.
Yeah, no, agree. And sometimes you have the right approach, but then the modality may be a different question. Do we go with a small molecule or a gene therapy, or what other modality might be more effective as well? I don't know, Deborah, you're going to make a comment. Just the question around blood-brain access and transport. I mean, the question is, how much do you need, and when do you know what exactly you're after, right? Because you can do a lot of nice experiments in mouse and maybe work our way through primates, but how do you understand that connection between what do we eventually need for the optimal efficacy? How do you get to know that answer?
Yeah, well, that's obviously something that there's a lot of active research on. I mean, if we knew the answers at this point in time, I think that a lot of our biologics approaches might be more effective. But there's no doubt that there is and I think to Tim's point, there are systems that actively remove aggregates from the brain as well. And these are sometimes the mechanisms that are also used to transport biologics into the brain. So there's this kind of paradigm where there's a flux between two kind of competing systems, or at least competition for the same receptors. So there's no doubt about it. It's extremely complex. I think biomarkers do play a role in giving you more information around the amount of biologic, whether it's an antibody or other kind of biologic that you need to get into the brain. But again, does the CSF adequately represent what's happening in the brain? I think is another question as well that clearly we're working on to see how we can fine tune that balance.
Great, John. I know you had a couple of other thinking as well, because as you develop, your agents, which are obviously non-A beta, non-tau, how do you think about that future as the questions are being addressed with certainly the A beta as well as the tau mechanisms? What do you see the other mechanisms play, particularly in the broad context of neuroinflammation? Perhaps you can start and then we'll go to Jesús. Go ahead.
Well, So I actually don't view ourselves as being distinct from amyloid and tau. I think it's in AD, I think neuroinflammation is just fundamentally the third leg of the story. With Rudy Tanzi talks about this all the time of that it's amyloid tau inflammation, and there's a rich interplay. And there is in the science, and that certainly we've been working on, and a lot of other people have been working on. If you look at Bill Mobley's work or Ralph Nixon's work, there's a very direct connection between amyloid, the inflammatory pathways, and tau. And it's that intersection is certainly where our focus has been. And there's I'm not going to go into the detail. I'm happy to talk to anyone about this afterwards, but it is, in the endocytic stress pathways, it is all in the synapse and linked to axonal transport, and all of these mechanisms are and deeply linked to proteostasis defects, where the aggregated proteins ultimately come out of. Right. And I think we should. And that's where where the biology is going, and I hope that goes and plays into this thinking about how the balance of these various proteins, including TDP-43 and other aggregated proteins, where they play out in any given patient. But I agree with Tim that his upfront comment. These are exciting times. Because the fundamental, the aducanumab approval and all the A beta antibody data that we've come out to, I think it validates fundamentally the disease pathogenesis model that we've been working on in AD, but it's not. I fundamentally don't think it's just for A beta. It actually more broadly validates the model, and within that model there are rich set of targets we can think about and work.
Yeah, and maybe just following up on that, Jesús, if you could maybe dial this back a little bit, and maybe we move away from our AD hat, think about other diseases as well. You're active in ALS, certainly MS. You touched on that.
Absolutely. Neuroinflammation, defined as the compartmentalized inflammation within the brain, mostly driven by microglia, is absolutely key. Not only to Alzheimer's disease, but to a lot of other neurodegenerative situations. And one of them is multiple sclerosis, where we see that the microglia turn into aggressive phenotypes, which attack myelin over time. And that's a process which is very well differentiated from the acute inflammation or adaptive immune system, which is also contributing to the relapses that we see in patients. But that today can be very well controlled by existing antibodies, including, for example, the anti-CD20s. Now, neurodegeneration, the key thing for us, at least, is to try to understand why is there some neurodegeneration. And this is why we believe that these proteins, which are encoded by patients and are able to actually activate microglia, they're able to interfere with the remyelination process, are really key to the long-term neurodegeneration. And we've done it. So we've done already a first phase 2 trial in MS, where we've seen that by inhibiting those proteins, we're able to very strongly reduce the levels of atrophy, the number of black holes, but also maintain myelin integrity by being able to stop that interference with the remyelination mechanism. Working with the NIH with another protein of this human endogenous retroviral proteins, which is dose-dependent toxic on motor neurons, which was also found in sporadic ALS patients. And it's all about why is there neuroinflammation, and we believe that there's endogenous retroviral proteins, which again are produced by patients, are key. And what is fascinating today is to see all the research that is being done on the neuropsychiatric symptoms that are experienced long-term by COVID patients. Not talking about the recovery from the disease in the first few weeks after it, but we have a tsunami of data showing long-term neurodegeneration in those patients, with the first signs also of atrophy, with the first signs of all the symptoms and effects of neurodegeneration. So that's why it's critical for us, at least what we're trying to do, to understand mechanisms that are pushing to that compartmentalized inflammation and try to see what we can do to actually stop them.
Thank you. Thanks very much. But would you like to add anything more to that? I know you're thinking also more about multiple sclerosis, and obviously, some strong tie-in with neuroinflammation in the broad context. But as you think about the next generation approaches in that space, what are the challenges you face with respect to advancing new compelling mechanisms in the sort of the neuroinflammation space? So obviously, with challenges on understanding the different animal models, tying it with human disease, the translational piece of it, all of that. Is there any different thinking that you or within your company happens that with respect to how you think about neuroinflammation broadly?
Yes, certainly. So we covered a lot of the neuroinflammation side of things. I think the other piece that is worth calling out is on the neuroprotection, which is another area where there is an area of active research. And I think Tim alluded to it right at the beginning. I think there is a lot of potential for combination therapies, which I feel haven't been explored to the same full potential. And you can think of scenarios where you have like an anti-CD20 therapy combined with a neuroprotective agent to really come together in either a form of a regimen or other fixed dose combinations and really make a difference. Again, I'm maybe not the expert to speak through the animal models and exactly how, beyond the traditional models and how your EA models, for example, would end up translating into humans, but that has always been a challenge, and especially in the neurodegenerative disease space, where it's very hard to have that full correlation between even the primate models coming up into the humans just because of that unknown with the blood-brain barrier.
Got it. And maybe one final question before I hand it off to my co-chair here. So as you think about, I guess, the non-A beta, non-tau approaches in the case of Alzheimer's or broadly, in neuroinflammation or other sorts of approaches that are being taken, how do you think about sort of prioritizing where you want to go? Because, certainly an area of biology is cuts across, different neurodegenerative diseases. And, what's the process by which you go through in terms of prioritizing and sort of selecting the indications? How do you want to try first and then, work your way through understanding more of that? I don't know who wants to take that on. Maybe John, do you want to start? Or Deborah?
I can. I think it is easier today than it was even 5 years ago. But I think it goes back to the point that I made earlier, that to think about the underlying pathology. I think where we're coming to in CNS diseases, we've been years, like oncology was just thinking about lung cancer as dementia. And, the initial sets of drugs, they work remarkably well in small cell lung cancer. And that differentiation has made a huge amount of difference as you get to different subtypes. In our case, where we've ended up with dementia with Lewy bodies is because it's a pure disease of the basal forebrain cholinergic system. And the biology around impaired NGF signaling and neurotrophic factors and the effects of inflammation on that are in the preclinical models are on basal forebrain cholinergic neuronal degeneration, where we've shown that our drug will actually improve that. Yeah. Now, we kind of stumbled onto that, but it actually comes together very nicely. And I think going forward, that's exactly the way to think about these things, is that what systems within the animal models that affect. And for example, in AD, early onset Alzheimer's and DLB are actually more similar than early onset and late onset, because they're pure diseases of the basal forebrain cholinergic system, and late onset Alzheimer's disease is primarily a hippocampal atrophy disease that in many contexts has TDP-43 and vascular pathology that's driving it as much as the A beta component. driving it. And I think that's the way we need to start. And we finally have the tools to actually think about that, of aligning the mechanism and the preclinical data with the specific outcomes we're trying to achieve in specific patient populations.
I know, Deborah, you had a point on that as well. Would you like to add?
Yeah, so we've become really interested in lysosomal storage disorders and looking more and more at the underlying biology and the genetics of these conditions. Of course, they're rare genetic diseases, but if you look at these as a continuum, so where there's haploinsufficiency in progranulin expression and amount in the brain, then that's leading you into frontal temporal dementia. It certainly leads to activation of the inflammasome and release of interleukin-1 beta. So we're really looking at that nexus between what is happening with the genetics that leads us into the interesting rare genetic disorders, but then looking at that continuum and looking at ways that we can help a broader patient population, such as frontal temporal dementia and ALS through replacement with progranulin. Yeah.
So it's biology and the pathophysiology that sort of drives that trajectory.
Yeah.
Got it. Great. Understood. Yeah, great. Tim, over to you now.
Yeah, we thought for the second half of the panel, so last third, we'd sort of cover more sort of trends in partnering, deal making, and I guess trends in the science as well. I mean, the first area we wanted to talk about was, I guess historically most companies have been focused on, small molecule and antibody, blockers or activators of various kinds. But there's been this explosion in the embracing, I guess, of more complex advanced modalities, from the gene to the RNA to removing the proteins a different way. So we just wanted to get your kind of thoughts around this. So maybe we'll start with you, Bharat, on a sort of pharma perspective. Would any reasonable shot on goal be okay for your company? Do you have any preferences? How do you go about assessing the sort of risk reward of these various things?
Risk reward is exactly how we start out looking at the new modalities, right? Because with a lot of these new modalities, what you are doing is you are taking on the modality risk, you are adding that on the biology risk, and that just ends up compounding. And one way of mitigating that is that reward sufficient, and reward especially in terms of the impact. Is this in a space with significant unmet medical need, and how can you try that together? Look, Merck's been around for 350 years, and the only reason we have survived that long is because we have always been looking at these next-gen modalities and trying to see where that next wave is. I would say that at this point, at least within our NNI group, we are starting to explore some of these. I wouldn't say we have left the boat with the small molecules and the MAVs to just focus on the newer modalities. We've definitely started exploring some of those. mRNA is one that is really a hot topic right now, but even beyond that, pluripotents and hemopoietic stem cells is one that we've been trying to explore. But I would say it's still the initial stages to see whether there's something that works in that area.
Right. I don't know, Murali, I don't want to put you on the spot after chairing the first half, but did you have any thoughts from the pharma perspective?
No, it's a good question. I mean, traditionally, we have all been small molecule brought up, and then biologics came along the way we became biologics companies. And the modality is an important feature, and the modality matters, right? I mean, it could be a great modality for a mouse, and you could have a nice, beautiful Nature paper, but at the end of the day, if you want to have a therapy for humans, it's a different question. So I think you have to look at a lot of aspects with respect to modality, and especially with new modalities. I think about ASOs, gene therapy, CRISPR, and others. There's a lot of unknowns. And I think the general approach that we would take is let's start dipping our toe in the water, see what's going on. Either it's with early investments we make, early collaborations, and depending on how much we learn, we can take a deeper dive at it, and maybe even form broader partnerships. At this point, we are not sort of setting up modality shops within the company. It's going to be driven by the targets. So if the target needs a modality, we go find it. And then if it's the right modality, then we find the best partnership to take that along forward. Either we bring it within the company or we partner closely to move it forward, because that expertise does not rely on us, it depends on somebody else. So it is case by case, but again, it's based on the target, the biology, the disease, what you want to go after. And of course, there are commercial considerations as well, right? You don't want, to be too invasive in a way that you may have a great therapy, but nobody wants to use it, although it may be effective. So there's a lot of balance and a lot of trade-offs we have to consider.
Maybe Jesús and then John. I guess if you found a super hot area of biology, would you take on a brand new modality to tackle it in the most productive way, or you think there's probably other paths with more traditional modalities first?
We're working on a very exciting new biology already. So if you combine new biology with experimental ways of delivery, I think that you're starting having an equation with too many unknowns to get anything that will be any interesting out of it. We're trying to minimize the unknowns. So we're going with well-known products, which are antibodies, and our main challenge is to find the right dose. And we're going on top to what Barrett was saying before. We're going on top of an anti-CD20 to really focus on the anti-neurodegenerative effects that we're having at relatively high doses, because we know the challenge of getting it into the brain. And it is hard when you're having new biology to also have new delivery systems and new delivery and new things. Again, it's you, when you're doing drug development, you want as few unknowns as you can in your equation. And for me, for example, in our what I believe is a very hot area of neurodegeneration, having at least as possible unknowns is really my goal.
So, John, did you want to add to that?
Yeah, I think, I smile like Jesús did in some ways. I am perhaps just old school in some ways. I'm a small molecule drug discovery and development guy, though my entry into the industry was leading the AbbVie clinical development program in MS, which ends up being perhaps for years the exception that proves the rule in terms of targeting the CNS with a biologic. And that was, the assumption was that as it is, that it's the peripheral immune system that's driving the disease in MS, which is what allows you to have the effect particularly in the relapsing remitting population. I think the untapped biology continues to be, at the end of the day, and this is, I would go to the amyloid biology, and which reaches into base as well, which I think is intrinsically part of the amyloid hypothesis, as well as tau. We need to get inside the neuron, and we don't have any easy ways to do that and get across the blood-brain barrier to this point without small molecules. And so I think there are lots of opportunities, and certainly in some of the specific rare genetic diseases where we know the specific biology, where I think gene therapy and knockdown strategies can actually do better than be more specific than small molecules can be. But I think in the complex diseases such as Alzheimer's, DLB, et cetera, that it is and age-related neurodegenerative diseases in general, that we're going to have to find a way to get inside the neuron and correct the fundamental defect.
That probably brings us really nicely on to Deborah. I think sometimes it's easy to forget whatever type of modality we're working on. This stuff needs delivering into the brain effectively and have the right biodistribution to the right cell types. I guess there's been quite a resurgence in excitement around delivery technologies per se, along with the biology. How are you finding that?
Yeah, I think that's absolutely correct, Tim. There has been a resurgence of interest in the delivery technologies, and particularly if you can deliver biologics that are already validated to a certain extent. So to Jesús's point about reducing risk, some of our work actually revolves around already approved molecules, but just delivering them across the blood-brain barrier more effectively. And through, as I said, just trying to open that window, we get more in. We know that, for example, when we're trying to deliver an enzyme, that the delivery system also goes to the lysosome within neurons. So, we're ideally targeting where we want that enzyme to land. With another payload, of course, within the brain, within the different cell types, the payload will land in a different place. But we certainly know with enzymes that are targeting the lysosome that our XB3 technology will not only get these enzymes across the blood-brain barrier, but also there is delivery into the lysosome. So we know that we're hitting the target there.
It's a great point.
Maybe to close this out, I mean, this could be not either or. It could be even a life cycle play, right? You could probably have an antibody approach. Once you know that, then perhaps there's a gene delivery approach that can maybe vectorize the antibody, deliver it appropriately. So, there could be stages and evolution of modalities as well.
Yeah, certainly at Lilly, we've invested in both sides, both the biology and modality, but also for gene therapy, different AAV vectors. We've looked at lipid nanoparticles, we've looked at brain shuttles, and also, ultimately you have to get it across the blood-brain barrier and or to the right brain region and cells, and probably need both of it ultimately. Great. Well, maybe for the last bit, we'll talk a bit more about the market, the money, I guess, and financing. I mean, CNS drug development is often long and expensive, and it really needs great partnership to be successful. It's rare that any one company can do everything completely alone. So we thought we'd cover the partnering aspect first. Maybe John, we read that there's a huge amount of money around. Everyone's excited. Is there still
just
a
hasn't
space
come by. It hasn't come my way. I just
me too. Is there still a lot of pharma interest? Yeah. I mean, obviously we read that companies want to go it alone a bit longer, but is there still a place for effective pharma partnering? How do you feel them? I mean, it used to be maybe many years ago, people could wait for maybe clinical POC data, but these days it's a very competitive space. So I just wondered what your thoughts were around the sort of general environment in CNS partnering.
Well, I think that, so it's obviously it's very different than 5 years ago. There's a lot of interest, a lot of discussions. I think the challenge, both from a partnering standpoint and the investor standpoint, for particularly where we are, is that in the pre and peri proof of concept in the clinical stage, it remains actually challenging. That dip in financing from early stage that it's been described as this black hole in some ways of that the getting to proof of concept, it's gotten, if anything, more challenging in some respects. Just purely from a financial standpoint, being able to manage that. I will say that in all of our pharma discussions, it can be remarkably refreshing to work with, speak to pharma who knows neuroscience and knows drug development. It's always clear how much small companies such as ourselves can learn from the experiences in companies like AbbVie and Lilly and Serono, EMD Serono, and it is and so we're absolutely there. But I think that pharma continues to be a little challenged in how to evaluate at this intersection between science and clinical. There remains and it's particularly acute in neuroscience and neurology, and it mirrors the academic world where there is a relatively big divide between neuroscience and clinical neurology. Remains that of the people who are doing each of those. I think Europe's actually a little bit better in that respect. But it's that gap of who you're talking to in a program that is deeply rooted in the science, but that is actually has proof of principle data in the clinic and is ready to move into late stage development remains challenging. And we're working on it.
Mm-hmm. We'll ask for the pharma response in a minute about whether we struggle in that zone. But thank you, John. Maybe Jesús and Deborah. I mean, I think both of you guys are also working in the preclinical space. How have you found the environment?
Well, actually, not preclinical. We're finishing off phase 2 at the Karolinska, right?
My project, yeah.
So, it's I completely subscribe with John's points about the importance of interacting with pharma and having an external view on your programs. I think that has always been extremely valuable for us. Whenever we can do it, we do it. Partnering as also is a double-edged sword, because we started a major phase 2 with 270 patients with a pharma partner, and the pharma partner decided that neurology was not fun a few years ago, when a lot of companies decided that it was too much of a challenging area. And then you're left with much less resources and have to be really smart in order to try, as I was saying before, to lower the number of unknowns in your equations and really focus on what you need to do. I think that in terms of the money, I really don't see a tsunami on money in neurology, at least not in proof of concept, as John was saying, and particularly not in Europe, where, as you know, the number of specialized funds in biotech has basically all but disappeared over the last few years. But I'm still gung-ho about the perspectives that neurodegeneration offer us. I think it's a tremendous unmet medical need, and original approaches, if you can demonstrate that they work, will indeed, find all the resources, whether it's pharma, investors, third parties, etc. What I have been very happy about, and this is also to John's point, is the tremendous support we get from the academic community, which it's basically they are looking at new ways of understanding and of treating neurodegeneration, and there's a tremendous amount of demand and also support that we're getting from, for example, the MS community, or today the neuropsychiatric community for COVID-19. They're basically doing all the work there, so to understand what's going on. So that's also a very important thing for us.
Great. Deborah, before we're running out of time a little bit, but just briefly, how are you finding the general environment?
I think it's been pretty well summarized by John and Jesús. I think early stage partnering, though, for conditions where there's no effective treatment, where very significant unmet need exists, and where there's an opportunity to really change a paradigm, there is still interest in early stage partnering.
Right, and I'll just put Bharat and then Murali on the spot very briefly. Do we struggle with early stage partnering? How do you view the current environment and the sorts of priorities that you guys want to execute on?
Yeah, no, I think John touched on it. I think it's that sort of very proof of concept space where there's a little tickling of activity going on. You got some EEG data, you got a little bit of that, and you just don't know how to piece it all together to say, is this robust? Is this proof of concept enough to invest in a larger, full-blown trial? I think that's a place where we all struggle. And I think part of the challenge is we don't really understand and have the connection between those signals of early pharmacodynamic efficacy or pharmacodynamic readout to the ultimate efficacy profile. I think that's a place we all struggle. And again, I think John hit it on the head. I think whichever mechanism you go, that early signals really doesn't give us that confidence to say, let's now invest in large full-blown trials. I think that's kind of where we all get caught up and we say, let's wait for a little bit more information, a little bit more data. And I think we get caught up in that little circle.
Bharat, did you want to add to that?
Look, agree completely with that. That is that gray zone, that dip where we need to find that right balance. And the solution really is risk sharing, having those discussions and having those partnerships, because this is a really risky area of development. And the more we can communicate and speak to and manage those risks, that's the best way forward. I do think there is a renaissance in this space. Like if 6 months back, you had told me that GSK would be re-entering into this particular space would have not believed you, but then they made a big move with the deal with Lector, with potentially over $1.5 billion on that and 700 million. So definitely this is a space that is really active. I think in a larger setting, the last 18 months of the pandemic has also put a highlight on healthcare as a space. So maybe all of that money is not there yet in NDD to the same extent, but I do think it is coming. So I am pretty optimistic of how the next few years will pan out.
Great. Well, I think we're out of time. So thank you to Bharat, John, Jesús, and Deborah for all those insightful comments. I mean, my summary is it's a challenging area, but there's a lot of excitement and enthusiasm for a whole multitude of factors, breakthroughs in science, compounds moving through the clinic. So thank you all for your great comments and for joining the panel. I'll hand back to Murali in case you've got any last comments, but this has been really super. So thank you, everyone.
No, thanks again. I think you summarized it very well. Thanks, everybody, for jumping on the panel. And we look forward to seeing you all live during one of the conferences in the future.
Hopefully soon.
Pretty soon, hopefully.
Very soon.
Great job, guys.
Maybe see
that. Thank you, everybody.
Right. Hey, thanks, everybody. Thanks for jumping on.
Thanks, everyone. Bye-bye.
Yeah. See you probably in Boston if many of you get to see Tad. But see you down there.
Yeah, I'll be there.
All right, we'll see you there in person then. Thanks again. All right, bye-bye.
Bye.